If we look at another avirulence factor, where we know the cognate R protein, so this is RPS4, we know a lot about the genetic requirements for death below this. We know it needs a gene called ADS1. I'm not going to go into that. Its function is actually still unknown. And it requires the phytohormone salicylate to get death. And what you can see for RPS4 is that we can decrease death. So this is basically ion leakage. And as cells are dying, they leak ions. So this is the wild type. You're getting an HR response. You're having a lot of death. Whereas this is EDS1, which is absolutely required for this cell death. And this is ATG7. So basically, this would tell you that autophagy is required for this type of cell death. So let me just go to the mutant, because in a Arabidopsis you can never have too many mutants. We isolated a long time ago ACD11. It doesn't really matter what it does, what this gene does, okay? Although if you're interested, I can discuss that later. It's a recessive lethal, and it has this runaway cell death. It induces many, many classic defense responses as it's dying. And its requirements for death execution are rather interesting. It requires, for example, EDS1. It requires salicylic acid, just as we saw for the HR. We can suppress death in it completely if we remove salicylic acid using a transgene, but we can kill it again, even if we've removed SA, by applying an analog of SA called BTH. And just very briefly, I'll show you that actually, if we induce death in this mutant, then we see activation of, in this case, autophagy 8. This is a classic biochemical assay for the cleavage and lipidation of ATG8, and also we get very nice decoration with autophagosomes. Okay, so this is showing all of the hallmarks that I just showed you for an HR response. So if you're a geneticist, you try to be smart and lazy, and you basically set up screens where the plants do the work for you. So this is an ACD11 plant that carries the NAGI transgene. It has no SA. It's perfectly normal. It has a nice life and many babies. Okay? On the other hand, if we spray it with BTH, we kill them. So what we did is we mutagenized these plants, and we did a very large-scale screen of 3 million M2s. And basically, after spraying with BTH, almost everybody dies, except mutants, or we could call them putants, okay, putative mutants, where you've made mutations in genes required for death. And we call these genes Lazarus genes, not because we are very Christian in my lab, believe me, okay, but because Lazarus was apparently some guy that Jesus brought back from hell to explain to all of the other sinners how bad hell was. And that actually fits pretty much with life in my lab. So these are called Laz mutants. And I'm going to tell you just briefly about 2 of them. I should say early on we got out known mutants that would suppress salicylic acid signaling. So we knew the screen worked. And I'm just going to tell you about Laz2 and Laz5 because I think they're somewhat interesting. So this is just, for example, showing you that the Laz2 mutation fully suppresses the mutation in ACD11. So this is how you identified it. Laz2 was known earlier as a histone 3 lysine 36 methyltransferase, which is kind of a mouthful. But this is basically an epigenetic regulator of chromatin states. Okay? And indeed, these plants are, for example, early flowering, and that's because actually Laz2, or what's called SDG8, is a regulator of repressors of flowering. Okay, and this had been previously known. So the reason that the way Laz2 works is that it trimethylates lysine 36 on histone 3, and trimethylation, if it's, depending upon where it is, but if it's in upstream regions of genes, including their first introns, then it's actually an activation mark. And so what you're getting is expression of the target gene that is controlled by